It's been a while. Before that, he studied engineering at Case Western, and his career has always been innovative with new ideas to improve the quality of his patients' lives. Dr. Rizakis.
Thank you. Thank you very much. Thank you. So, yes, I'm an ophthalmologist. I guess we all are clear on that. About 10 years ago, I became interested in anti-aging medicine. So I've been doing bioidentical hormones for about 7 years. Then about 3 to 4 years ago, I became very interested in the field of methylation, and now I'm lecturing to you on nutrigenomics, which is, I find, a phenomenal field of great interest. I have to acknowledge my co-presenters. Brian Bakke, he's my mentor in this. He's a PhD in bioorganic chemistry. He came from the food industry as well, and Brian is also an expert in methylation, so he's my mentor in methylation. A lot of what you're going to hear today is thanks to Brian. And then Vasily Rizakis. Vasily conceptualized a very unique idea that I'm going to show you in a moment on how you can very easily bring nutrigenomics into your practice. So we are all involved with an entity called xrmd. That is our financial disclosure. Maybe some of you came to our booth today. So what is nutrigenomics? It's the first question. It's the relationship between diet and health. And I believe it should include nutraceuticals as well, or medical foods, as part of the concept of the diet. Then the question is, well, what is health? I like to think of health as optimized gene expression, optimized biochemical pathways, and optimized endocrine function. I think epigenetics, which is what I love, is health optimization, short of changing one's DNA code, which apparently they're starting to do now, which is absolutely incredible. I think nutrigenomics is a subset of epigenetics. So I don't think nutrigenomics includes bioidentical hormones, stress management, acupuncture, physical exercise. Maybe some of you disagree. That's fine. We can talk about it afterwards. So in this lecture, I want to take you through the basic science of gene-nutraceutical interactions, the clinical relevance of nutrigenomics, how genetic testing can influence food selection. I'd like to touch on inflammation and nutrigenomics, and how you can combine hormones, nutrigenomics, and get clinical outcomes. Let's talk about methylation and gene expression. So as many of you know, we are built of 5 billion code letters defined at the moment of conception. That comprise 30,000 genes and 23 pairs of chromosomes. And then the Human Genome Project came along and taught us that we're not perfect, even though our mothers tell us we are. We have errors in the spelling of our genetic code, which are called single nucleotide polymorphisms. So why does that matter? Well, it matters because if you have single nucleotide polymorphisms, it's going to cause a problem within your biochemistry. So what you need to do is think about genes and biochemistry. So here's mom and dad. She gave you a genetic code, but that code codes for an enzyme, and that enzyme does something, right? It takes A and B and produces product X. And if all is perfect, if mom's perfect and dad's perfect and the code is perfect, then the speed of this enzyme will be normal, 65 miles per hour. So guess what? If you've got a misspelling in the code, as shown by that G instead of a T, you've got a problem with the integrity and quality of this enzyme. So product X will now be produced at a slower speed. Isn't that fascinating? I love it, because it has a lot to do with the methylation pathway. The methylation pathway is enormously important, and it's shocking to me that it's not utilized in every discipline in medicine. It impacts my field: macular degeneration, diabetic retinopathy, glaucoma, cataract. Methylation is basically everything and everywhere. There's 19,000 articles written on homocysteine, as shown here. Just go to PubMed and type homocysteine, see what you get. I think it's around 20,100 articles now. So why is there so much interest in homocysteine? Well, the answer is because it has to convert through these enzymes here to methionine, so we can create SAMe, so we can methylate many things in the cell. And those many things in the cell include biochemical reactions and your DNA. So when you methylate DNA or a gene, you shut it down, you control it. So gene expression has a tremendous amount to do with what you see right in front of you here. And that's why MTHFR is so important, because if you have an MTHFR dysfunction, you're not going to produce enough 5-methyl-THF. This methyl group will have a hard time getting flipped onto B12 through MTRR. You're not going to make enough methionine, you're not going to make enough SAMe, you're not going to methylate, you're going to become a hypomethylator, and then you're going to get sick. How? What disease? I don't know. But something's going to happen. And if you are a hypomethylator, somebody in your family is probably sick, and you can pick it up in history very easily. There's 7,000 articles on SAMe, about 6,000 articles on MTHFR. And there's even more to this wonderful pathway, because look, right over here is glutathione. There's 42,000 articles on glutathione. Everyone in this room knows glutathione matters. Your cells have to produce it. But there's an enzyme right here called CBS, which actually manages the movement of homocysteine down to glutathione. And you've got VDR, which has to do with vitamin D3 receptors. So if you have a genetic error there, you have a problem. Now, with the colors you see, I apologize for not introducing that. Green means that mom and dad gave you a great set of genes. Yellow means that mom or dad gave you a misspelling in that critical gene. Therefore, the pathway will be broken there. Right? I mean, we've known this pathway for over 100 years. But now we know where to look in the pathway to see where it is broken. And by knowing where it is broken, we can then fix it. And that's what's exciting. So a burgundy color is a double SNP, which means it's mom and dad gave you a problem. Here's monoamine oxidase A, but look, here's serotonin, dopamine, and norepinephrine. Now we're in the world of depression, ADHD, and other behavioral disorders. And hiding out over here in the corner, minding its own business, is DAO, which is diamine oxidase. And that has an important, has a lot to do with histamine. So I'm trying to set you up for the vocabulary of the remainder of the lecture. I want you to become alert to the word SAMe. Here's SAW, S-A-H. One SAMe methylates something, it breaks down into SAW. Then SAW has to drop off adenosine and come back to homocysteine. And this happens in every single cell of your body. It is core to normal cellular physiology. Okay, so we can fix this pathway. That's the good news. It's all good news. When you do these tests on patients, you celebrate the results. I've got good news for you. You've got a BHMT problem. I need to give you trimethylglycine to push this pathway faster so you can methylate. Maybe you need SAMe, maybe you don't. Depends upon the clinical situation. You don't just routinely give SAMe. You need to give it only when needed. For example, if a patient has elevated SAW, you don't want to give SAMe because SAMe's got to break down to SAW. So there's an art to this. I hear people talk about nutraceuticals, try this, try this, try that. I don't like it because it's not rational, it's not clear. It doesn't make sense. You've got to start with the genetics. If you don't start with the genetics, you're kind of lost, quite frankly. So a nutraceutical here is methylfolate, methylcobalamin here. If you have an MTRR, of course, if you've got a VDR, you need to give vitamin D. And you can give quercetin here to help DAO. And you can give molybdenum here to help SUOX break down sulfites. So nutraceuticals are part of the therapeutic approach to managing the methylation pathway. Let's talk about gene expression. Let me go back 2 slides. If you have a problem with MTHFR, MTRR, BHMT, or some combination thereof, homocysteine is going to elevate. Okay, that's why there's 20,000 articles on elevated homocysteine. What that's reflecting are abnormalities of these 3 genes right here. Okay? So hyperhomocysteinemia is a sign of genetic error. It's a sign of methylation pathway disturbances. You got to understand that, otherwise the rest of the lecture is going to be meaningless to you. So if you have elevated homocysteine, you've got a problem. You probably have an MTHFR, MTRR, BHMT. Okay? So now, look at this. We're talking about gene expression. Gene expression is part of nutrigenomics. Well, if you have elevated homocysteine, it can induce the downregulation of 52 proteins and upregulation of 44 proteins. Well, that's gene expression. There's something wrong with the methylation of the gene. Let's go back another 2 steps. Remember, when you methylate a gene, you turn it off. If you're not methylating it, it's turned on, so it can be overexpressed. So if your methylation pathways are not working correctly, your gene expression will not work right, and proteins will be produced when they're not supposed to be produced, or vice versa. So elevated homocysteine is telling us that there's a problem with methylation, there's a problem with the control of the gene, and this paper is saying to us that some proteins go up, some proteins go down. That's gene expression. Tau protein is an important one, isn't it? Because of Alzheimer's disease and chronic traumatic encephalopathy. Well, that's interesting that hyperhomocysteinemia will actually impact tau proteins. I thought that was really interesting because now you're talking neurodegenerative disorders. Let's talk about nutrition. Nutrition matters because if you have a CBS polymorphism here, that's the sulfur gene, okay? Now, unlike all the other genes you see here, if you have a CBS polymorphism, the enzyme is working too fast. So then it's going to overmove homocysteine down to cystathionine and work its way down, produce ammonia, other problems, etc. Unfortunately, it doesn't produce excess glutathione. You typically have some oxidative stress as a result of that, and Dr. Bakke could explain why. I'm not exactly sure, but the bottom line is CBS means sulfur-containing foods will go rushing down this pathway and cause a problem. So if you have a CBS polymorphism, you may have a problem with some symptom. Could be migraine, very commonly, could be others. So nutritionally, what you do is you tell that patient, do not eat foods that contain sulfur. Okay? So you can use food as a medicine as a function of genetics, nutrigenomics. Actually, it makes sense. Now, here you see BHMT, trimethylglycine can help push that pathway faster. And so you have to tell the patient to eat foods that contain trimethylglycine. And the food that is fantastic for trimethylglycine are beets. So you might recommend beets or simply give trimethylglycine as a nutraceutical. Here are folates. You can help this pathway work better. But if you've got an MTHFR, even if you're eating well, it's not going to help because you need the MTHFR gene to work properly. But if it is working properly, nutrition can benefit the production of methylfolate, which will help produce methionine, which will help produce SAMe, which will help you methylate, which will help you control your DNA properly. MAOA, that's the tyramine pathway. MAOA has to do with the ability of the cell to break down serotonin. If you have an MAOA problem, you're not breaking down serotonin that easily, so you need to produce less of it, and you can do something about that by restricting your diet and foods that contain tyramine. So that's that pathway. So here's tyramine, here's sulfur, and then there's DAO, which is histamine. If you have a DAO, you've got a problem breaking down histamine in your gut, and as a result of that, when you eat foods that contain histamine, like me, you feel sick, and that's a bummer. But I'll tell you one thing, if your patient has migraines, some of you I'm sure have already figured this out, a histamine restrictive diet can benefit quite a number of migraine patients. It's one little facet of migraine. Having said that, I want to make the point right now that you don't fix a disease with one therapy. You've got to fix a disease with multiple attacks on the disease. It's not one answer. It's multiple answers. It's multimodal. It's combinational. And that's what's so vexing about being here at this meeting, because everyone's trying to figure out what combination of things must I use to treat that disease. And you've got hormones and nutraceuticals and food and all this. So that's the challenge of this, is the multimodality of it. But anyway, here's nutrition. Histamine, you've got tyramine, you've got folate foods, and you've got beets and sulfur, as an example. So there they are again, sulfur, tyramine, histamine, trimethylglycine, and then dietary folates can benefit. Clinical correlation. We've been, as I mentioned at the beginning of this talk, I've been doing this now for 4 years. So we've had the luxury of treating everything on this page here successfully. It's benefited the care of patients. And the beauty of this type of care is that it's valuable across multiple disease states. So we've treated ADHD patients, acne, anxiety, arthritis. We've benefited them. Okay? We've benefited them with this nutrigenomic concept I've presented to you thus far. So that's literature and clinical experience. And then you have simply the literature. The literature suggests that what we're showing can benefit these conditions as well, but I don't have anything clinical to really fall back on to say, yes, I've seen a case like that. And I hate doing that too, because I hate the anecdotalness of things. We need to do studies and more studies and show how all of these modalities can actually treat 50 to 100 or 1,000 people, so we can have real numbers and figure out exactly what combinations of things can treat that disease correctly. That's what we need. Well, let's go through the literature. So in red, you know what that, you now have it, right? You see SAMI, you know what SAMI is, right? Homocysteine, methionine, SAMI. So here's literature in the field of neurology talking about the relationship between SAMI and Alzheimer's. Here's SAMI and SAW with Alzheimer's and elevated homocysteine. Remember, there's 20,000 articles. They're easy to find. Here's another one with homocysteine and Parkinson's, PSP, dementia, ALS. Here's a great paper that just came out. It's a review paper, nice paper, talking about elevated homocysteine, which you now know is a marker for bad methylation, and how it has a direct toxic effect on the vascular and nervous system. And look at the diseases that they indicated is involved in: heart, atherosclerosis, MI, stroke, minimal cognitive impairment, dementia, Parkinson's, MS, epilepsy, and eclampsia. See, this is the fun part of this, because when you're down at the genetic level, we don't know what disease you're going to get, but we know that if you have these genetic errors, it's going to worsen many different clinical conditions. So it's not surprising, okay? It's not surprising. It's like air. If I take the air out of this room, we're all going to, have a problem. But the point, it's not a great analogy, but the point is, the point is that, this pathway matters a lot. Okay, here's an interesting fact. I think everybody should know this, that if you have a patient who's on levodopa, interesting enough, levodopa will actually increase homocysteine levels and actually worsen Parkinson's. So anyone who's on levodopa needs to have their methylation pathways checked and treated, because it will increase homocysteine. So that's important to get out. Migraine is a very simple slide because we've treated many migraine patients. We looked at our results. We have a 92% success rate in really, really tough migraine patients that have failed Botox and have failed the drugs, failed Topamax, etc. And when you go at these patients with methylation and or hormones or both, you can really do a great deal of good. So that's that. If you go back to the literature again and look at autism, ADHD, and learning disturbances, there are multiple references on this where elevated homocysteine, MTHFR, hypomethylation, glutathione issues are part of the pathogenesis of the disease. So I'm clearly telling you that we need to check methylation pathways. Let's go to the world of gastroenterology. Here's SAMI again and its relationship to colon cancer. Here's SAMI with inflammatory bowel disease. Here's SAMI again with gastric cancer. So again, you see the connectivity between methylation and disease. Here you have relationships between SAMI and colon cancer. And here's a paper talking about how 1-carbon metabolism, which is methylation, is related again to cancers in the GI system. So it's published. Rheumatology, there's literature connecting methylation to lupus, connecting methylation again to lupus and RA, and connecting SAMI and methylation to tumor necrosis factor expression and chronic inflammation. In the field of cardiology, you all know this, that if you have elevated homocysteine, you're going to have a greater risk of heart disease. And again, here's literature to support that. Elevated homocysteine, problems with SAMI and SAW, and vascular disease. Okay? And there's more and more here. This paper almost threw me off my chair when I found it about 3 years ago. It talked about cancer, and it said, in particular, global hypomethylation, which basically means problems with the methylation pathway, is the most common DNA lesion of cancer cells. I thought this was absolutely incredible. That hypomethylation is a precursor of many cancers, not just any one or two. It's pretty much all of them. We could talk more about that because some of you are thinking, what about hypermethylation of genes and the like? Right, you're shaking your head. Yeah, you got to hypomethylate first before you hypermethylate. I'll show you a paper about DNA methyltransferases. It's if you mess up the cell, it goofs itself up, produces too many DNA methyltransferases, which then shut off certain genes and they look hypermethylated. So you would think don't methylate patients because it could cause cancer. That's not true. Hypomethylation precedes hypermethylation. It took us like a week or two of debate to figure that out, so there you go. Psychiatry, the folks at Harvard are using methylfolate to treat intractable depression, but that's insufficient because you can't just give one thing, you got to look at the entire pathway. And then based on the entire pathway, you can round it out, and you can do a lot for patients with depression. And obviously, why would you ever want to put somebody on an antidepressant as a first line of therapy, given the side effects of antidepressants? I mean, give your patient a break. Try this first. It doesn't have side effects. If maybe it doesn't work, then you can try your antidepressant. Usually you don't need it. Because I don't like taking people off of Effexor when they come to see us, because that's hard. It's very frustrating. I'm sure many of you know that, to wean them off of Effexor and some of these other antidepressants. So it just makes no sense to me that anybody should be subjected to an SSRI or an antidepressant until they've had their methylation pathways checked and treated. What's this? Here's a paper, serum homocysteine levels with behavioral and psychological symptoms of Alzheimer's. Here it's talking about elevated homocysteine being associated with symptoms related to Alzheimer's. This is a big one here. I had a great meeting today at the show with an OBGYN doctor. She came to me and she said, I need somebody to tell me that there's a relationship between autism and methylation. I said, there is. You're right. I've looked at the literature. It's disgusting how clear the literature is on the relationship between autism and methylation. It's disgusting because no one knows about it. It's published. No one's looking at methylation. It's just terrible. So if you're going to have a child, or if your son or daughter is going to have a child, check their methylation pathways. This is a good one. I didn't expect to find this, but MTHFR actually is associated with Down syndrome. I was intrigued to find that. So it's not just autism. It's you got to be on top of that. Here's another paper from China. The Chinese do great work, by the way, in this field. And they looked at MTHFR and MTRR, or that other one, and found it was associated with complex congenital anomalies. Oh, that was interesting. We've treated patients with dermatologic disturbances, and they did well, and that reflects the literature, which says that they should do well. And here's another paper connecting homocysteine MTHFR with psoriasis. Okay, let's talk about inflammation and nutrigenomics. There is a relationship, is the bottom line. If you have methylation pathway problems, you can have inflammatory problems. Some of the prior speakers were talking about inflammation is bad. Every meeting you probably went to at the show here talked about inflammation being bad. Okay, great. If you have methylation pathway disturbances, that can be a problem too. So here's a paper showing how 1-carbon metabolism or methylation can be associated with C-reactive protein elevation. Okay. If you have a person on statins, if they have elevated homocysteine, it can interfere with some of the anti-inflammatory processes or properties of statins. I don't like statins, but nevertheless, you don't want to have elevated homocysteine if you're giving someone a statin. So those patients need to be looked at. This is a great paper which connects homocysteine elevation and inflammation and shortening telomeres. So this is anti-aging. Right? IL-6, many of you know more about this than I do, but it stimulates inflammation and autoimmune processes. And there's a connection between homocysteine elevation and IL-6. And here's that paper I was alluding to earlier, talking about how inflammation causes DNA methyltransferases to cause hypermethylation of other genes. But nevertheless, bottom line is hypomethylation precedes hypermethylation. Here's an interesting paper which I liked. It talks about IL-6 being released because of histamine. And I said, wow, that's interesting, because SAMe, remember I said to you earlier that SAMe methylates 40 different chemical reactions in the cell? Well, one of the reactions that SAMe methylates is the breakdown of histamine. So if you have low levels of SAMe in your body, you're going to have a harder time breaking down histamine. Well, you know how interconnected everything is. If you have a problem with SAMe, you can't break down histamine, so you're going to have a release of IL-6 and cause inflammation. It's a nice little fact. I thought that was intriguing. And DAO, remember what we said, is the enzyme in the gut that breaks down histamine. So if you can't break down histamine, your body will be inflamed. If your body's inflamed, it's going to cause inflammation, and one of the mediators of that could be IL-6. Thought that was interesting. Clinical results. So let's keep it simple now. So what we do is we look to see what foods cause inflammation in the body. Because I think I've made my point that inflammation is a problem. It can impact methylation. and gene expression, which connects ... to nutrigenomics. This is all still under that umbrella, nutrigenomics. We haven't left the mothership. And so food, first thing I'd like to do is see what foods are causing inflammation. So we run this test, and we determine what foods are causing inflammation in the body. This is IgG related. And the green means they're okay, gray means that there's some inflammation, and burgundy means there's a lot of inflammation. That's a lot of data, right? Now, remember what I said earlier. I told you that your genetics will also tell us what foods are not good for you. I told you about tyramine, told you about sulfur, I told you about histamine. Well, here's a patient who has an MAOA, so they have a problem with tyramine. This person has a DAO, they have a problem with histamine. Lucky for them, they don't have a problem with CBS, so they don't have a problem with sulfur. So now your mind has to juggle all the foods that are related to tyramine and histamine and reference that and cross-correlate that with all the foods they're allergic to, right? Well, fortunately, Vasily here, one of the co-authors of this paper, said, why don't we put this all together? So he and Dr. Baki, who came from the food industry, who's got a PhD and he's an expert in methylation, they got together and they produced this report. So this is a report that we create, I call it Nutrigenomics Made Easy, that tells you what to avoid and what to eat in moderation. And we try to color code for you why we say what we say. So the black type here are foods that are bad for you genetically. In this case, you know it's tyramine and histamine. And the blue type means what foods are not good for you from the standpoint of allergy or inflammation. The prior speaker was right, it's all about inflammation. So food sensitivity only. And then there are foods that just have an issue with genetics and inflammation. So we color code that in green. Bottom line is, these are the foods to avoid. These bigger numbers mean eat them in moderation, but maybe once a week. Here's 2 to 3 times a week. And these greens are pretty much okay. So what do you do with that? If you've got a healthy patient and everything is fine, you tell them, well, you better be careful about those foods, because if you eat them, you might not feel well. And they say, yeah, but I really love egg whites, and I really love chicken, and I really love... Well, if you eat them and you don't feel well, don't blame me, because I'm telling you that might make you not feel well. That's one patient. Next patient comes in, and they've got migraine, irritable bowel syndrome, depression, and other problems. That person, you say to them, eat the ones down here. Eat the ones that are not going to cause inflammation inside your body, and eat the ones that are not going to upset the apple cart of your methylation pathway. So that's Nutrigenomics Made Easy. Now, here's where it gets really fun, is when you combine what I just showed you with bioidentical hormones. That's the mothership. That's what happened to me 4 years ago, after doing bioidentical hormones for 6-7 years. You know, in walks Dr. Baki, and now we're doing methylation. And then when he and Vasily figured out how to integrate all this food data, then we went to town. And that's why our migraine results are so good. I quoted you a number of about 92% success, is when you integrate all these things together in a multimodal, combinational manner, the results really do go up significantly. In fact, you know what's really cool? Dr. Baki told me this, hold on a second, at the end. All the questions will be in the back at the end. You know what's really cool about this is you don't need as much hormone when you treat a patient's methylation pathways. We found that, so as a result of that, we actually treat methylation first, and then we give hormones. Because you don't need as much. Because you know that old expression, if all you have is a hammer, everything looks like a nail? So if all you have is hormones, then everything you throw at people are hormones. And here you can do more. We've treated ADHD patients. This is more with the nutrigenomic portion. And those kids have seen improvement. We should do more studies on that. I've seen lupus patients improve dramatically. I'm not surprised when prior speakers talked about their experiences and they put lupus up on the chart. I agree. I don't know if you care about retinitis pigmentosa, but we found we could at least get some vision back from some of these patients. Arthritis, yes. We've been able to get patients off of Enbrel and these other biologics. And then children, you know, they need this. Why would you want to saddle a child and not treat their methylation pathway disturbances or let them eat foods they're allergic to? so you can make the kids healthier. This is a great case here, which thanks to Uzi Reese, I was able to figure out what went wrong. He brought a cardiologist over to talk to me. This is a patient who was, he had a myocardial infarction. He had some heart block. His heart wasn't firing. It would beat too slowly. So his pacemaker would click in 14% of the time. And we put him on this program, and his pacemaker stopped working. In other words, it didn't need to work anymore. I couldn't figure out why. And I'm going to tell you why later at the end of the talk. Depression, yes. Of course, there's literature to support that. We've seen bipolar patients improve. That was quite dramatic. And it was only when we gave them a methylation program that they improved. Ankylosing spondylitis, that was an amazing case, had the lowest level of methylfolate ever. He tricked us, though, because he had a normal MTHFR gene. And we measured his methylfolate levels in his body, and they were ridiculously low. So sometimes some of these patients can trick you. They may have a normal gene, but their biochemistry is off. He wasn't absorbing when he needed to absorb to make methylfolate. So when we finally gave him the methylfolate he needed, he started improving. And various rashes tend to succumb to methylation pathway, as shown by the prior literature that I showed. Nutrigenomics is crucial for cellular health. I want to make that point here today. You know, the concept is this is cellular. The disease is not the disease, it's the cell. It's not the leaky gut, it's the cell in the gut. Okay, it's the cell. Whatever the cell is, I don't care, it's the cell. When our cells become sick, we become sick. It just depends on what cells become sick that determines the chronic disease. This is the way to think about what this is about. So this graphic here is what we basically do, is we're treating cells. We look at the genetics of the cell, we find out where the polymorphisms are, then we know where the biochemistry is broken, right? And then we fix the biochemistry with targeted nutraceuticals to work around the abnormalities in the biochemistry. And we use the same genetic information to figure out what foods not to eat based upon genetics. And we also add in the inflammatory component, right? And so here's the biochemistry part, methylation, here's the nutritional part. And then, of course, we can use hormones if they're needed. And this is why that person's pacemaker doesn't work anymore. Apparently what we did for that person is the cells in the sinus node, apparently, that are responsible for beating the heart. I guess we fixed the cells. They must have gotten healthy, and they started working properly, and he doesn't need his pacemaker anymore. That was explained to me by a very, very smart cardiologist today. So I finally figured out why, or was explained why. Because I couldn't understand it. So why would his pacemaker not need to work anymore? It's just all about cells. So, you know, medicine needs help. That's why we're all here. The practice of medicine is obsolete, wasteful. Eric Topol, I'm sure all of you have seen this. But the answer is to treat the biochemical, nutritional, hormonal, and genetic cause of disease, and in the process, lower the cost of care and prevent disease. And I am done. That's it. Thank you very much. And feel free to contact me if you want to talk more about, if you want access to some of this technology. I can't talk about that here. We can do that in the hallway. Thank you. Have a great day. I hope you enjoyed your show.